is about bringing the disciplinary science with the big data and those who are interested in complexity to see to what extent we can bring better solutions to the challenge at the convergence between health, healthcare, and economics that we are still facing. So within that context, I am very pleased to welcome the participant to this webinar, and I'm shifting to Jay DePass to introduce the center as co-organizer of this webinar.
Hey, Brett. As Sean briefly introduced me before, I'm Jay DePass, the director of the Public Health Applications Group at Carnegie Mellon's Supercomputing Center, the PSC. Our generalized high-performance computing resources to non-traditional applications in public health modeling, modeling tools to new and broader audiences for national and international public health decision support. I'm presenting both the PSC and the MICE mission group that the PSC is a member of, which is networking and education arm of the Informatics Services Group funded under the Models of Infectious Disease Study, MIDAS, by the NIH. And this is a collaborative network of research scientists who use computational, statistical, and mathematical models to understand infectious disease dynamics and dissemination to prepare for, detect, and respond to infectious disease threats. So the goal of the mission group is to allow software developers from national and international network of MIDAS research groups to work together and learn about new technologies and practices. And Bridge webinar series in particular kind of pushes those boundaries to things that are a little bit more outside of the run-of-the-mill infectious disease modeling stuff that we do. So very interested to hear us talk, which I'm sure will fit that bill. And I'll hand it over to Sean to introduce himself and today's speaker.
My name is Sean Brown. I'm the Associate Director of Research Software Development at the McGill Center for Integrated Neuroscience, Neuroinformatics. I represent the Healthy Brains for Healthy Lives initiative, which is a co-sponsor of this webinar, which is a very large $84 million Canadian First Research Excellence Fund grant to McGill to improve the lives of Canadians by advancing understanding of how the individual brain functions in health and disease throughout our lives. And I have the absolute pleasure of introducing not only a very great researcher, but also a colleague for many years, Gary Ann. Gary Ann is a general surgeon with an expertise in surgical critical care trauma surgery and acute care surgery. In addition to being an active clinician, he is senior fellow of the Computational Institute at the University of Chicago. Diane is a founding member of the Society of Complexity and Acute Illness, SCAI, and both past president and current board member of the Swarm Development Group, one of the original organizations promoting the use of agent-based modeling for scientific investigations. Dr. Ann's research looks into harnessing the power of big data towards better surgical care. He has worked with on the application of complex systems analysis to sepsis and inflammation since 1999, primarily using agent-based modeling. His research involves the development of mechanism-based computer simulations in conjunction with biomedical research labs, high-performance parallel computing architectures for agent-based modeling, artificial intelligence systems for model construction, and community-wide meta-science environments, all with the goal of facilitating transformative scientific research. And on a personal note, when I first got into agent-based modeling in public health, it was about the time that I met Gary, and I have to be very honest, Gary's views and seeing his presentation on how to use modeling and system science in areas that I've never even dreamed it was possible to use it in really inspired me and my work. So I consider Gary one of my sort of mentoring researchers to really mold my views, my current views on how we can use these types of approaches to really inform health and well-being of people. So without any further ado, I will turn it over to you, Gary, and thank you very much for agreeing to give us a webinar.
Very much. Thanks to the organizers, and Sean, thanks so much for your very kind words. So Yeah, it's my honor to participate in this bridge webinar, and as mentioned earlier, this is almost a science of philosophy-esque type of talk. I'm sure they'll hopefully have lots of discussions about some of the things I bring up, but I look forward to interacting with you all. So, of course, my titles unfortunately turned out to be quite a mouthful, so I'm not going to bother rereading it. However, we're going to give you all a trigger warning because as a surgeon, I have to show you at least one gross surgical picture, and it's the next slide. So those of you with sensitive abilities, you can avert your eyes. The reason I show this particular slide, let's see here, because it keeps me grounded in what inspired me to start doing this research investigation research, because most of my career has been spent as a trauma surgeon, and that's a very kind of blood and guts type of thing with a very tactile, tangible, short-term, reward-oriented practice. So here is a picture of a fellow I took care of 8 years ago now, who was stabbed at the base of his right neck. It lacerated a major vessel that goes from the base of the neck into the chest. And what you can see here, a picture of the operation where the patient's head is to the screen right, and the patient is opened up to crack the ribs to kind of create a trap door, and the lung is sitting there, that kind of modeled thing that's sitting in the chest. And we've the abdomen open to release pressure, because we didn't have any injuries in his abdomen. The amount of blood that we had to give him and fluid, he swelled up. So this guy required about 70 liters of blood product and resuscitation within the first 24 hours. For those of you who know, the human body holds 6. So this patient had over 10 times, 11 times his total blood volume replaced in the 24 hours around the time of this injury and this operation. And the fact of the matter is that as impressive as some of those numbers may sound, or dramatic, or TV worthy, the excellence of this procedure is basically just cutting and sewing. It's super complicated. It's stressful, but it's not really complicated. And doing this procedure in the middle of the night, I'm thinking and realizing that the primary determinants of this patient's course are actually completely out of my control. So bleeding, I can give them the blood back, but the of molecular events that have been triggered by this injury and our care for the patient is going to cause a severe disruption in overall biological and physiological homeostasis, leading to multiple organ failure, subsequent infections, delayed recovery, and a whole series of molecular processes that we have absolutely no control over. So fortunately, this fella was able to survive. Eventually needed a kidney transplant because his knees were so shot from this particular injury. But at some level, it's very, very, very frustrating because you can what you can do, but the fact is that you have very little control over the vast majority of what unpathophysiologic processes that are leading to his potential demise. So when I think about these sorts of things, I always try and wonder whether or not there are fundamental issues to what we can know or what we can't know. And this is manifest in 3 challenges that have kind of been circulating around for the last decade or so that I think represent exactly these types of fundamental questions for which complex systems analysis and computational modeling can be applied. So the first, the translational dilemma, what I call the translational dilemma, or popularly known as the valley of death of drug development, and I'll talk about that in a second. There's more recently this issue of the crisis of reproducibility, which is essentially epistemic unreliability with respect to what is published in the literature. And then kind of the goal of what people purport want, with developments in biomedical therapeutics, which is or precision medicine. So it's going to be, kind of pejorative to how precision medicine is actually used, at least in the United States, because the phrase means a very specific thing, to, for instance, in the cancer world. But I'm going to use more of the kind of Webster, what the words mean definition, which I would interpret as the right drug for the right patient at the right time, and act towards being able to do exactly that. Right. So my own research goal is to try and develop robust fundamental approaches that can potentially bind these dilemmas together. And this is kind of my underlying philosophy of what science is supposed to do, which is be able to take knowledge and generalize it in a way that I can tie different types of phenomenon and say what I learned in this particular circumstance is applicable in this other circumstance. Right? And exactly these three challenges. Right. Dilemma, the Valley of Death. There are multiple iterations of this type of graph. Shows a divergence between what people spend and the number of drugs that actually come to market. Right. 8, 10 years ago, there might be a little bit of a blip, morally increased entities, but at least in the United States, it's arguable whether or not that is actually due to increased efficacy or laxening of regulatory structures just because we're so terrible at actually doing this. And what the Valley of Death represents is the transition from preclinical research to clinical implementation. That, the entire bioresearch workflow that looks at candidate targets, the molecular and cellular understanding of the pathophysiology through Proxy models of cells and experimental organisms. You come up with great ideas, and then you try it in people, and it just doesn't work. And it is important to remember that every single failed drug passed the entire preclinical workflow component, and the rate is what's called the Valley of Death. So this is a problem. So why exist? So at some level, this is a multi-scale problem. It's associated with the organizational complexity of biological systems, which I've demonstrated as a series of nested eggs on the left side of this screen. Think of this multi-hierarchical organizational structure that's inherent to biological organisms. There are these epistemological boundaries that are present at each level, at transition from one level of organization to another. And this is, not an unusual thought when people think about categorizing complex systems. And the challenge here is that our investigatory workflows are very bad at crossing these particular boundaries. And at some level, as again a foundationalist type of thing, I think of how is it we actually get to know things. So we have this traditional scientific cycle that has served us very well, since the early 17th century with Newton, where we look at them, we collect the data, we identify relationships between those data, make inference with respect to how those things come about. Those are hypotheses of inferences of mechanistic causality, and then we test those hypotheses with experiments. This is essentially how we know things from a scientific basis. Right? Every piece of technology that we have is a byproduct of this thing. So, but it doesn't seem to be working, because why else do you have the Valley of Death? So at some level, it has to do with the fact we've been able to deal with our low-hanging fruit diseases, like public health things related to hygiene and stuff like that. So we try not to draw our drinking water from where we use sewage and such things like that. And the diseases that we're now dealing with, like cancer, sepsis, autoimmune diseases, wound healing, are actually disorders of complex systems as opposed to single order external perturbations. One of the challenges has to do with the embarrassment of riches associated with the imbalance in our technological advancement. So we've significantly increased our technological capability to create experimental objects, extract data from those objects, and actually push correlations of those objects through advances in various aspects of technology, including high-performance computing. The problem is that in this data-rich, and we have an imbalance in the scientific cycle. We still require a very serial, semi-archaic process of evaluating our suppositions of causality, our hypotheses. So where we think of the data generation and data analysis as being effectively parallelized and augmented with technology, we still rely on, lab test graduate students, the lifespans or fragility of biological organisms to test those. So this is a problem. So the fact of the matter is that this cartoon represents the current operational strategy in pharma, where we just kind of hope that we picked the right target in some way, right? 9.99% failure rate of drug identification to clinical approval kind of does the free nature of this particular problem. Right, it's an issue. But it's gotten worse in the last couple of years because all these types of scientific workflows are predicated upon a reliable or trustworthy repository of knowledge. And the question with the crisis of reproducibility. So you may have heard of this term recently in the popular press. A lot of it is related to aspects of psychology, but this is also true in the cancer research area, where experiments that are the foundation of extensive lines of drug evaluation just apparently cannot be reproduced. Therefore, it has an economic impact on irreproducibility, and there have been consortiums aimed at trying to reproduce some of these in papers, and they're just not very good at it. So, there are a multiplicity of proposed causes. You can't go to any sort of general science meeting and not address this. And too often, blame is placed on the incentive structure, the publishing structure, the ability, the emphasis on positive results and not publishing negative results, and that kind of stuff. From disordered incentive structures or all the way towards fraud. And while those things are true, I believe that the majority of researchers are actually terrible scientists or actually, in the system in that particular way. So I think actually that there are fundamental components biomedical research is done that lead to this particular problem, right? I suggest that the crisis of reproducibility is due to a perpetual undersampling of the system's behavior space based on how we investigate that particular phenotype, those phenotypes. The denominator problem. So for this particular diagram, let's consider A, this irregular shape, to be higher possible state space of a particular system. And I'm thinking at a microgranular level, so not at atomic level, but by any sort of extracted molecular information you can get about this system. How do we investigate this system? Well, we sample population, right? So we try and get information from people and sample in some sort of way, but we realize that that's actually a very small segment. The key here is that the shape of A cannot be inferred to have a normal distribution, right? Because of a normal distribution or an assumption that a sample of a space can be inferred to represent that is based on a hypothesis that the central limit holds at some level, and we know that the central limit theorem requires independent variables, and these are not independent variables. So I don't think that you can make an assumption about what the shape of the distribution is going to be. We use experimental proxy systems like, rats, cells, etc. But the principle of a good experiment is limit the variability of what can be extract experiment so that you get a strong inference of causality as the result of that experiment. So the very nature of doing those experiments, you are restricting the space that you're examining with that particular object. And who's done laboratory work knows how fragile and sensitive to initial conditions these particular experiments are. You get the raw water source, you have a new postdoc, you change the air filters, you get a new supplier for reagents, and the entire system has to be re-engineered to produce the result that you have. Our fragility of any conclusion of an experiment created conditions would have to be assumed, right? Because the experiment is so fragile that every time you change one of these individual circumstances, it essentially begs for the crisis of reproducibility to rear its head. So, I assume the crisis of reproducibility is a fundamental property of how biomedical research is executed. So, what do we do to get beyond this? So, this, at some level, causes us to have to realize what biology actually is. And my argument is that biology is more treated like alchemy than like a analytical science. Right? It's almost empirical, and biologists make their name by expertise with respect to a very narrow slice of actual biology. It leads to the danger of infinite regress, which means just giving more detail about that system, and therefore I will know more about it. Little perturbations on those experimental forms. However, we're just trying to indirectly make inferences about what's actually happening because the entire set of mechanisms is opaque. Emphasis on components in biology, which is a historical artifact based on its legacy from zoology and botany, which are descriptive. There's no unifying processes, so there are really no natural laws in biology as we think of in either physics or chemistry, with the exception of evolution, which we can talk about before. But anyone should look askance at people who claim to be able to apply engineering principles to biological systems because the criteria for what constitutes an engineering problem often does not exist. So, big data is really powerful, and there's all this stuff in there about how big data actually works. And certainly, it forms an essential component of the scientific workflow. That part of the answer because it's only part of that particular cycle, and it can be a trap. And the reason that it's a trap is because it reinforces a human tendency towards liking magic, right? So there's a visceral appeal to the idea if you just collect enough data and you throw a big enough computer at it with a sophisticated enough algorithm that it functions like magic, and that you will somehow get a answer, as if you were going to the Oracle of Delphi, and it just tells you what, and but it's kind of a cheat because it obviates the hard work necessary to actually try and understand how things work. So, a simple example of what this data-centric categorization legacy can look like. So this is from one of my favorite authors, Jorge Luis Borges. And I find this particular classification system, which is actually not very different than any sort of metadata list associated with any patient population. And scale agnostic because it can deal with demographic features all the way down to gene expression data. I really like number 14, the one that from a long way off look like flies. So how does this actually impact how we know things in biology, or more importantly, how we argue about things in biology? So there was this paper that was published several years ago in PNAS. It's the output of the GLUE grant, which is giant. Hello? Looked at how well genome responses in mouse models reflected human data. This was actually, taught in the New York Times, and led to a lot of gnashing of teeth and rending of hair for the angst generated by this particular statement, because at some levels it purports the need to do preclinical models. An inevitable backlash for things. So a whole bunch of papers, some of which are very creatively titled to emphasize the difference between data set, but to choose a different clustering algorithm or means of analyzing this. Right? No way to resolve which one of these papers is correct and which one of you should follow, because it's really based on analytical method that was chosen, and there's no ground truth with respect to which one is better or which one is worse. Danger in trying to look at lists of components and trying to infer similarity between one or another within different contexts. Right? And the product of evolution, which has a tendency to reuse pieces that are put in one place for other purposes. The way to get around this is to focus on functions, not components, because functions are those mechanisms by which those components interact, and the means in which one phenotype arises from another. And we should always think about biological systems as dynamic systems, and therefore places an emphasis on trajectories through some sort of multidimensional space, but there be no supposition of path uniqueness from one point to another point. Right? This is a property of biology that arises from the fact that evolution occurs. Because if this path uniqueness, diversity was purely dependent upon heterogeneity within micro state, well then every mutation would be no basis for diversity. So since there is a relatively finite set of things that biological systems need to do to survive, these functions are actually conserved, and therefore I would argue that when we think about biology, we need to keep in our mind that we are thinking about functions, and that the components we identify need to be appropriately contextualized, and the data that we extract represent snapshots of those functions, unless you have a mathematical or dynamic computational model. So in kind of like cartoon set theory version, what we're seeing here is a description of what the traditional biological experimental workflow is, where you take in vitro and you something out, and then you try it in vitro, and then you get to clinical, and this is how it works. And the dashed lines represent inferred knowledge. That's an injective relationship if you're thinking in terms of sets. The target of the arrow incorporates a whole bunch of stuff that may not actually be present in the deliverer of the arrow. Right. So for this is what is the justification of the translation of the knowledge, and for what is actually similar and conserved as you move through this particular workflow. Paradigm number one, which is kind of depicted in both that paper and the Borges satire, is the idea of looking at components as the means that bind from one system to another system. A certain amount of validity in that at a very high level, but when we get down to the details, it is very, very uncertain. Right? Partial functions at best. Biomodels are opaque. We don't actually know how they do what they actually do. That's what we're trying to find out. Alternatively, if we think of the functions that are present, be that metabolism, homeostasis, movement, apoptosis, main tissue architecture, those particular properties are actually conserved across different systems. Right? So what we do is therefore identify what is the nature of the injective function, what that function that is conserved from one context to another context. That this is the role of modeling. Right? The modeling produces an explicit injective function because you've written your model. You know everything about it. It is no longer opaque. Therefore, the relationship between the mathematical or computational construct and the biomodel is an explicit thing. It allows you to have sound truth, at least in the left side target of this relationship. Modeling is therefore to build a hierarchy of models. Since they're models, you know that the relationships between one and the other are explicit, therefore completely transparent. And match these with biologics at these various levels, and you use the models themselves as a binding agent to translate ability from one context to another context. Right. So that looks fine in concept, but unfortunately in practice what has happened is that the criteria for what constitutes a trustworthy model becomes too rigid. Now there's a reason for this, because of course you want a relatively high fidelity in your models, but also from other domains in which modeling paradigms are applied, those domains which are primarily the physical sciences have natural laws that can constrain and ground and provide a high degree of truth to whatever the simulation is actually done. Right. So one of the dangers now is if what we consider to be a good computational model requires high fidelity to a particular biological model for which we extract information out of, i.e. a experiment, the bioproxy model can't serve as a bridge, right? And they're no longer connected to each other, or have to be vastly re-engineered to be able to bridge cross contexts. So this is a feature of modeling going forward. Right. A quote from a fellow who knows things, apparently has a good history of knowing things. For this particular paradigm, which is again a very natural sort of thing, challenge that we need to overcome as we think about how modeling and simulation can be used as a pathway towards theories. Right. And this theories represent the output of the science, that is what binds things together, identify what we consider to be similar, was fundamentally the problem associated with the three challenges I mentioned at the beginning of this talk. So we think then what is actually observed by biology? What is the range of phenotypes we see? I would say a parameter space of the functions that are being embedded in particular models. Right, so by heterogeneity from conserved structure, we know this because individual people can manage different phenotypes through their lives. Twins are not exactly the same. And who's done cell culture clonal experiments realizes that the cell population, even the clonal population, does not behave the same to this perturbation. And the breadth of phenomenon or data represented by a particular model is more important than the fidelity to a particular constrained portion of behavior space. Right? I call the importance of outliers. So let me example. So if you've done an experiment with a bunch of mice, say you've done 5, and kind of clustered around the thing that you want, and then one is kind of far away. As well, what happened? Example is, well, you did something wrong in the experiment. Well, I don't throw that out because some that was not approved or whatever. But you know what it is you did wrong to cause that particular outlier. You can say with all certainty, is barring measurement error, is that biological object is able to produce that particular output. And that information is extraordinarily important if what you're trying to do is extend the explanatory power of any sort of hypothesis that you come up with. Right. So thinking about biological heterogeneity as a manifestation of parameter space causes us then to be able to move towards this idea that if we use models and simulations of various components or aspects of these biological systems and we explore those parameter spaces with high performance computing, that will get us a better idea of what the actual denominator is. So we're talking about, so this is the diagram from here. A space, B is what you can say sample clinically, see what you sample experimentally. If you Proxy model, right, you believe, right, and we're not about what constitutes the basis of that belief at first, but just assume you want to investigate that. That vastly explored at a vastly greater scale and level of granularity than any other proxy system you can have. Argument for HPC augmented modeling and simulation exploration as of addressing the denominator problem. To address the denominator problem, if you can encompass all these different outputs and phenotypes within the same explanatory structure, that gets you to addressing the fundamental nature of the crisis of reproducibility. I mentioned I'm not a math person, so I learned how to do agent-based models because you don't need knowing math to do these. For those of you who are not familiar, agent-based models are object-oriented, and they look at a system as a series of interacting components that have can be grouped into classes that have rules, some stochasticity associated with how those rules are executed, and the actions produces population behavior, which is analogous to system behavior, which allows you to see the consequences of these rules in population. That agent models are the most general type of model. We can fight about that at the end if you want, but I would propose that it can encapsulate virtually all other methods of mathematical and computational representation. Most act with agent-based models by either going through the movies or playing video games. These battle scenes in these movies are CGI'd ABMs, where each creature is an independent computer program, and with what are originally thought to be very realistic stand-and fight rules. However, when they first started doing these simulations, they actually couldn't get the armies to fight. They would come in contact and then they would turn around and run away. And this example of how population effects supersede what an intuition says is a very reasonable agent rule. It's a demonstration of where the whole is greater than the sum of the parts. And this is very important in biology because our basic biological methods are examining how cells behave. So as you can cross this translational divide, essentially reverse Humpty Dumpty, the process that we use to distance apart to investigate them, which you have to do, right? No other way to find out what things do other than the reductionist scientific component. Right? The problem is aggregating that information in a way that reproduces the context and the complexity of the original system. The agent-based models are excellent modalities for doing this. Now that we're thinking about using models as modes for investigation, to this particular paper, which I think is probably one of the most important papers in the last 10 years, and this from PLOS Computational Biology in 2017. It is a study that asks, can a neuroscientist understand a microprocessor? And this paper did, as it says, well, we have this information processing object, a microprocessor, for which we have ground truth because we've made it. We've engineered it. We know how it works. We know everything that goes on inside it. But we look at that object as it were opaque, as it were a biological system, and apply to it the methods that we use in neuroscience to try and understand how the brain processes information. What meaningful things can we actually say about that system? Just how tractable are current methods up there, and it's that you actually can't say anything meaningful about how the microprocessor functions. You could take these input-output matrices, right, that say, okay, you do X to the microprocessor, it does Y, but it can't tell you how the thing actually works. And the paper, of course, is that, we know how a microprocessor works, but you can't do that with the experiment unless you have a conceptual model of how you think that microprocessor works. That then represents a different investigatory track that gain the increase understanding. Right. So, propose to use agent-based models as experimental objects to determine the limits of analysis and current methods to investigate disease. We a couple of years ago, as a result of a NIM-BIOS workshop, thinking about the place of agent-based models in this particular investigatory process. Because we can do this maybe at the end, but if you can come up with a more formal mathematical model of a system you don't need an agent-based model for, then you should never bother making an agent-based model, because they are, like biological systems, opaque to how they do what they actually do. Right? With respect to what goes into them, but they produce the behavior that they have, there's no formalism to describe that. So as agent models are generally treated as experimental objects that you observe, run, and evaluate. So they're taken there, but with high-performance tuning, you can just do that in orders of magnitude greater than you otherwise could. With a Sean alluded to, I've been making agent-based models for quite a while. This whole list of them, but we'll focus on the ones related to sepsis because that's kind of the problem that my fellow got stabbed at the base of the neck had after we fixed him up. This is essentially a disordered systemic inflammatory response due to usually infection, but it can be due to trauma or hemorrhage. Kills more patients every year than these other disease processes combined. It's in some ways the first disease that reflected the translational dilemma, because back in the 1990s we had actually a pretty good pathophysiologic understanding about what the cellular and molecular mechanisms were. There were a whole bunch of preclinical drugs that were anti-cytokine, anti-pro-inflammatory agents that were thought could be successful based on preclinical research, and then failed utterly in clinical trials, and continued to fail over the last 30 years or 20. Then my first was models of sepsis, where we made exact representation of the human body as an interface between the line blood vessels and inflammatory cells, and the concept was that inflammation would break its containment and therefore spread into the blood, leading to a propagation of the inflammatory cascade, where you now had collateral damage to otherwise normal tissues. Separated the knowledge that was present at the time that the cytokine trials were developed. What can we say about sepsis with this particular model? And the first question is, well, how do we evaluate it? Here's a screenshot of kind of a wireframe diagram of the interactions, just to show that it's reasonably complicated, but not overwhelmingly so. This goes back to this idea about heterogeneity as a manifestation of parameter space. So think of that wireframe diagram as being conserved from one individual to another individual, and actually to some degree from one organism to another organism. This is that we actually see from patient to patient represent parameters of the relationships of those particular functions. Right? That's one way you can think about differential genetic predispositions, because those alterations in genes lead to subtle differences in the control response capability of those molecular pathways. So, to be able to do then is identify what is the state space, the functional state space associated with sepsis based on very high level population output characteristics. Another aspect that is evident when you start making this model is we are perturbing it. There are different factors related to the nature of the organisms or the microorganisms that we are purported to insult the system with. Right? These four external variables. So this resilience, which is how does the cells recover after they've ceased being damaged, and properties of the bugs that we drop to the system. So environmental contamination, representing different aspects. So do is see what is a realistic behavior space based on these particular properties. So start at the very minimal plausible behaviors with this. So the system can't always die. So if you drop one bug on it, this system might be so fragile that it just dies, right? Because then it would never exist. It can never die, which means that it has to be killed in some way, because that's how the real world works. The ultimate population dynamics that we're looking across a range of perturbation is that sometimes it heals, sometimes there's infection but dies, and this is what we see in sepsis. And so you have overwhelming infection, right? Because it's got to be killed. Every three broad categories, we're able to evaluate what is the parameter space that we can then use as a proxy clinical population when we start using this particular system. This is of the number of parameter sets that were evaluated. We ran a little over 60 million simulated patients, which is more patients than exist in the entire clinical database for all sepsis through history. These are the results. So this is an example of what is a plausible population dynamic, where on the left screen, with lower levels of injury, it is able to heal completely. In the middle, seeing where the inflammatory response is effective to be able to clear the infection, but otherwise dies from a forward feedback tissue damage. And then start to see overwhelming infection where the system is not capable of actually containing the underlying infection at all. Right, so this is a polar outcome. And you can imagine that there are very different distributions of this, but since we don't know what the initial parameters are in the real world, we have to consider that they're possible. So the space looks like, since we can plot three axes at a time, these are properties here and here with respect to aspects of the microbes, and here is the resilience of the system. And green represents those parameter sets that actually are able to produce clinically plausible realistic behaviors. Across that parameter set, we can consider that a possible representation of a human population. Now we characterize what we consider to be possibly biologically relevant. We now try and get more granular in, well, what can that tell us about the various trajectories or states that those systems, particular instances, actually can see? So the more mathy type of stuff. Or what we're trying to think of is there a formalism that would characterize our agent-based model and give us insight into the power of deeper analysis? So this is a random dynamical system. I think that's pretty evident. Since it's random, you don't have fixed attractors as you would in a deterministic dynamical system. So we come up with this idea of these probabilistic bases of attraction that represent, what the state space looks like in terms of what the possible future paths of a system based on its particular state space. Right? So we think of probability distributions. The analogy would be to think about the shapes of the various electron orbits are based on the Pauli model. And while these two-dimensional plots do have some structure, so out here you can say that these systems are going to die, right? All biological interest occurs in the rainbow section, where there's a great deal of uncertainty. And these are actually pretty big in terms of what the possible interpretation of future trajectories actually are. So that if you standard correlative hierarchical classification, you can't actually find any sort of state identification that predicts eventual outcome. So why is this important? This is important because This is what I'm going to do for 25 years. Any attempt that look at biomarker panels to predict outcome, be it cytokines, various omic scales, etc., none have predictive values much better than about 80% ROC curves, which is not good enough for clinical practice. The question is, well, why does that not be the case? And the trend is always, well, it's because they're hidden variables. It's because there's too much. We need data. Our granularity of description is not sufficient, etc. What we did now with this model is analogous to the computational biology model. Is to say, well, now we have a proxy system. It's vastly simpler than the real world system, but it's pretty complex. We have knowledge of this. We can get as much information out of the system as you want. So we're not limited by n. There are hidden variables. Given a system with this particular kind of multiple feedback structure, is it possible to come up with a classification system that predicts? And the answer is can't. Right? Some nonlinear classifiers to look and see whether or not there are combinatorials or second or third derivatives of some of these trajectories. But the fact is that the separation in any of between the blue and the red on PCA plots suggests that the marker predictive capacity is futile, and you shouldn't do that. You should do something else. Right? Forecasting component of sepsis. It's just that if you're focused on the data-centric or state space identification or snapshot-oriented approach to identify a predictive panel at one point in time, that's just not going to work. It's just not fundamentally plausible. But position, it's not good enough for me just to forecast, right? I want to be able to take someone who is going to do poorly and make them do better. And that back to the idea of precision medicine, which is the right for the right patient at the right time. And the reason sepsis is that there's no effective biotherapeutic after 30 plus years of research. There's no FDA approved drug for this. In the original publication of that sepsis ABM back in the early 2000s, where it tested a series of drugs that had in clinical trials and said, well, if you had this tool, you would have reason to believe that those drugs would not have worked. Argument that the reviewers came back with is, well, you have this model, what does work? And for 15 years, we were unable to answer that question because we were still limited by the same sort of inferences that you would be making if you were doing biology. However, with advances in computational methods, now have different ways of investigating control or controllability. And the attempt at this was to determine whether or not, if we use a genetic algorithm to search across a multidimensional multi-time point intervention space, could we converge with a solution that works? And what does it actually take? If we're not familiar with genetic algorithms, they're essentially a way of effectively searching a large parameter space using evolutionary principles. In this particular model, we had these however many cytokines that we could manipulate within the particular model. We used 4 inhibition strengths and 4 augmentation strengths. The combinatorials of that is just crazy. So we use a genetic algorithm which just samples across, take the top fittest, you jumble them around, you try it again, and you keep iterating until you get you converge to a solution. You can actually get a solution, right? What that policy looks like. So these are time points and intervals every 12 hours, second 12 hours, etc. These are variables that you can manipulate. Red, you inhibit them. 3 is you augment them. And it turns out if you do this policy that reflects, manipulating these 14 different cytokines in different fashions every 12 hours, you can take a mortality rate of nearly 80% and make it down to about a little 10%, which is pretty good. And it also suggests that this policy, this particular strategy, is effective across other parameter sets. Right? And the differential trajectories or dynamics that are present in the various regions of parameter space, as from that prior graph. So now that we've identified a group of non-responders, what is it about those non-responders? So that they have this property where they have an augmented granulocyte colony stimulating factor that happens to them. What that actually is, but represents a functional difference, a trajectory difference that's present in those particular patients that could represent the target of a evaluation at a particular point. It turns out you can train a therapeutic range for this particular group. Right, this shows is the prior therapeutic policy that produces this line. You retrain the model at that particular point, and you come up with this different policy that also takes the mortality rate down to less than 10%. With the GA, it's again something we identified doing these investigations. That's a stochastic zone in that model where the ultimate trajectory is almost random. So the idea then is that we need to be able to drive the system out of this, in other words, out of the attractor bases, right? So that then the system will either go one way or the other, ideally down here. This is called adaptive control. We're looking at different approaches towards this using control models that represent representations of deterministic dynamical systems. So another method that we have more recently talked about was deep reinforcement learning. Some of you may be familiar with this method. This is a machine learning artificial intelligence approach that in particular circumstances has been used to learn how to play games. This is a method that involves training a deep convolutional neural network, and you basically link it to a game where it looks at the position of what the board looks like, and then will play simulated games, which works out best. Most recently been in the news with respect to the AlphaGo or AlphaGo Zero system that is better Go than any human person, and again, it's a DRL trained AI that was able to do this by playing billions of simulated Go games against itself. So iterative learning process, but it requires a simulation of the system to train on, where you can't use static data, at least in this particular fashion. We propose to do that with the agent-based model as a proxy for sepsis, as a proof of concept. Because of concept, we gave it a great deal of latitude in what it could observe and what it could actually manipulate. It turns out if you train a system on this, AI on this, it can come up with a policy that has takes a 40% mortality and cures everybody, which is pretty impressive, because even accounts for the stochasticity of the system. Just as GA, we wanted to see whether or not this was generalizable, because the training set worked, and where we tested on other regions within that parameter space, that plausible parameter space that I talked about earlier. And it is pretty robust, because it took of these other 500 different initial conditions, plus these different mortality rates, basically cured just about everybody. The circumstance did not heal, worsened, which was one of the concerns. It is adaptive control. Each one of these colors represents the interaction that is applied to the system as a result of the policy. This one reflects the differential approaches based on different levels of the recurrent nosocomial infection. And this shows the exact same policy applied to 3 models that have different increasing levels of injury. Right? So the least level of injury in the system more. And it's clear differential trajectories in what the manipulations are going to be on the different trajectories that the target system actually has. We're in the process of connecting the observable and action space to approach more clinically relevant policies. Right. Of this talk is that beyond big data, we kind of need simulations for really big data. And this is what's done in the physical sciences, whether you're doing theoretical physics or you're doing meteorology with respect to weather forecasting and that kind of stuff. You have representational models and that it's produce probability distributions of possible outcomes at which you check your theories. The idea of clinical heterogeneity, which is the bane of an experimentalist's life, but is absolutely essential if we're trying to make any sort of translational concept, is a function of parameter space. And therefore, simulations to fill in these gaps because that shape B will never approach shape A. This will use data not necessarily as a source to derive our hypotheses. They'll use that at some degree, but at some level, the statistical power requirement, because now we're looking for the outliers to determine coverage of existing data. If you're going to really utilize HPC effectively and the advanced machine learning technique and evolutionary computing techniques, you need to do those on simulation, right? Because the people who do for real jobs, when they are fit biological data sets, they are substantially smaller than what is normally used in the index samples for machine learning. So a couple of summary thoughts. Biology needs theories. We need abstract representations for what concepts similar between one context to another context, so we can transfer that applied knowledge. I think 8 other complex types of simulation models are a bridge for biological knowledge. You can bridge these discrete experimental objects and individuals. We want to have forecasting and control as we do in other domains, right? To be simulation based, because those methods are reliant on that iterative workflow between analysis and simulation. So with that, I will finish up and take questions that you might have. Thank you.
Thanks, Gary. That was quite an interesting talk. I really appreciate it. I'll take the first question because I wanted to push a little bit on the idea that agent-based models are they can't. I'm not sure I'm trying to decide whether I fully agree with the idea that the simulation model is not. You can't have insight into how it's working. Are you referring to the fact that you set up the rules for the agent-based model, and then the stochasticity, the interaction of the rules is stuff that you haven't set up, and therefore, you run the agent-based model and sample over the system. There's no way to predict a priori what the rules are, the interactions between the different agents will be. Is that sort of the nature of that assertion?
It is that, because you actually do have a lot of information about what goes into that particular model, but the translation of those particular interactions into the future trajectory of any particular instance is extraordinarily poor. Right? There are essentially no equations of motion for agent-based models, And that means that if you're going to try and understand something about an agent-based model, you end up just having to run it over and over and over again, which, is not the worst thing in the world, right? Because it's better than the real world because we just can't do it at that particular scale. But that does represent an analytical gap between how we use agent-based models and how we may use other types of mathematical formalisms. We say the same thing about a very complex ODE. 150 equate ODE, you can't say about that either, right? So, and therefore, I would include, these very highly complex, multi-interaction, multi-feedback systems in that group. It would be nice if we could come up with formalisms to describe the equations of motions of those systems, but in the absence of that, we have to use simulation at a very large scale.
Okay. I will defer the next question to Lorette, who I'm sure is waiting to ask a question.
Yeah, thank you, Gary. It was a very, very inspirational talk for us that are struggling. I'm having in front of me your first slide on multi-scale translational challenges. You go from the genes to the organism, and in our brain to society, we are saying we need to nest the biology within society and understand behavior. So I guess you can have a sense of why we were so pleased to have you, because we do believe that there's a lot of learning to have in what we are doing from what systems biology has been doing. And I have a first question related to that slide that I have in front of me about the vertical and parallel coupling. If you could kind of talk a bit more about how should we think about building or thinking of conceptualizing those interfaces, whether it's vertical and parallel, of what the input of one does to the next level. And as you give the rules to your agent in some way, you need to have a sophisticated understanding of what are those interfaces that are happening in those coupling. That's my first question. And the second question relates to your last slides when you talk about the looking, and it's related also to Sean's question, this synergy between the agent base and then your precision prediction, which is, I think, is getting very much into understanding how can we build more synergy between the various AI and agent base and the statistical other methods of inference. If you could give also more address this last part also okay. Thanks.
Yeah, so for your first question, so obviously, as you well pointed out, there's an egg that goes beyond the organism, which is populations of organisms. That at some level, these organizations represent, let's call them evolutionarily defined modules that, for whatever reason, based on how evolution works, there are bow points where a bunch of process converges onto a selectable phenotypic unit that then represents the modality that produces the fitness function that evolution actually operates on. I think under the biology has done a pretty good job at characterizing coarsely what those actually are. So cells represent a very natural sort of encapsulation for a whole set of behaviors that happen at a subcellular level. And those are able to interact to produce cellular constructs that are histologically defined as tissues that are then organized into functional physiologic units, which are organs, etc., scaling up. The natural point the transition from organism to population of organisms is the organisms themselves. All right. The key is to remember that dynamics at some level represent constraints on what the solution space and the organizational structure is present for each one of these particular hierarchies. So the vertical coupling is essentially the transition from cellular to multicellular organisms, and then the population effects that drive addition of fitness and natural selection. And cancer in particular can be the devolution of what the evolutionary fitness function is for a particular cellular population. We'll leave that aside for a second. So that's vertical coupling. The parallel coupling is path redundancy. The idea that some functions, that the robustness of a particular phenotype and is a necessary process of evolutionarily determined solutions. It leads to redundancy in capabilities, not exact redundancy, but some mild diverse redundancy that can collapse into identifiable unit fitness function for a particular environment. Right? Because it was very brittle, that is efficient enough to rise to a particular fitness function for a particular context. But once you change that context minutely, that fitness function may not exist in. Therefore, there needs to be some play, some robustness in the type capability of those generative processes, and that's what the parallel coupling, I think, comes from. So, to your first question. To answer your last question, I think I would go back to this idea of the scientific loop. Right. So, to me, machine learning and the learning AIs, those are powerful pattern identification, correlation, structure identification methods. Right. And that says, given a particular data set, I can tell you this about that particular data set. But the trade-off is it can tell you only about that particular data set. It can tell you nothing about what happens if you do something to the underlying system so it could produce data different than the one that you actually had. Right. So to make an inference about what's going to happen if you do something different, which gets to control, you have to have some way to forecast what that is going to be. It cannot be done with a traditional correlation and pattern identification AI. So mathematically, what you can think about this as, assume that you have a system and you have it with a great deal of granularity. You can come up with a Markov transition matrix, absolutely predict probabilistically what a particular trajectory is going to be through that particular space. But there is a control on that system that now is what that matrix looks like. That matrix is useless. You can't think about it. Now, what can you do going forward? So I can do the correlated AI can do two things. It can fully characterize a particular data set or give you insight into that particular data set. And it could potentially identify hidden features of the system that is generating that data. This is what generative reinforcement learning does. Right? Stuff that comes out of these adversarial neural networks. Those can tell you what the behavior of the resulting hypothesis structure or mechanistic structure, causal structure looks like. It's actually simulated. And that's where the complex simulations come in. And instantiations of particular presumed causal structures actually generate what the behavior spaces look like. That loop. And then you go back and say, well, which one of these are plausible and which ones of these are not plausible. Back to Sean's question with respect to how much can you say about the behavior of a particular structure. All these complex enough that you can't necessarily infer or infer what the future behavior of that system is going to be. And therefore, any sort of assumption about what that population distribution of data or state is going to look like, you can't do until you simulate some sort of replication of what your underlying conceptual model is.
Thank you. I will offer someone else.
Yeah, more follow-up question, Gary, and I think one of the, and I guess maybe I want to make sure that I have this right, so you can correct me if I'm missing the point a little bit. The work that I've done in age-based modeling mostly involves epidemiology and population level age-based modeling. And we're always constantly fighting this struggle in the epidemiology world that people go out and do a cohort study. They do their statistics on the model, their biostatistics on the model, and come up with correlations, and then make broad statements about what that means. It's always found it interesting that in that world, it would seem like people believe that is a more accurate or more insightful approach than sitting down and coming up with an agent-based model that has been tested, iterated with knowledge, and incorporates some of that, but ultimately very testable and able to capture things that one cannot gather from a straight-on biostatistic study. And it would seem to me that those are complementary approaches rather than one being a gold standard versus another. I certainly wouldn't put epidemiological data collection studies as gold standards because you can't control for all the confounders that they're looking at. So a lot of our talks with decision makers, we try to get across the point that modeling is not necessarily it's not accuracy or inaccuracy, it's more about an understanding of the system and having that testability in the simulation model that you can't have in real life, and having the ability to be able to put further knowledge that one gains from these epidemiological studies in there to test to see how it may affect the population. Is that along the same lines as some of the stuff that you were talking about how you really relying on simulation models as part of the predictive process?
So that is almost exactly correct. So to recast the classical approach, which is empirical, is that what is the denominator problem? Whatever, if you have a particular data set, you analyze that data set. You that data set till the cows come home and be able to make very, very, very strong statements about that particular data set. Mm-hmm. But that's the question. What is the extrapolatability of that data set to a representation of whatever overall context you're going to use it? That's the denominator problem. So you have faith that data set represents that the generative processes that are present in that other system, and that those things are held, then okay, that's fine. But you can't make that statement because you don't know what those variables are. You don't know what all the factors are. Your conceptual model may be wrong, etc. Mm-hmm. Right? What people are coming with is empiricism associated with traditional research. They make this next step, which is not justified, which is the relation of that knowledge to a context that is not represented or may not be adequately represented by that data. For the other part of it, what I say is recognize that there is a insufficiency and unreliability with respect to whatever explanatory mechanistic model we come up with. We have knowledge that they're incomplete. And therefore, part of the process of using them requires the use of ensembles of models that represent a wide range of potential explanatory hypotheses. So I use the example of how weather forecasting goes, right? Because there's a set of forecasting models. I think there are like 20 or 30 of them that are constantly being run in parallel and then being updated with data feeds that determine how well certain models perform given real-world empirical feedback. And then you refine what that forecast horizon is based on updated information. ultimately how precision medicine has got to work, right? Because you need some representation in which to project a forecasting cone of potential trajectories into the future. That horizon is something that needs to be determined empirically, but you need to have that capability to draw that cone ahead of time. And therefore, and then this is the link that was talking about, where you link it to ongoing data updates and data analysis to be able to refine what that projection horizon is. And it's a random dynamical system. There is no other way. That's the only thing you can do.
Yeah. I agree. Cool. I will open for. Does anybody else have any more questions for Dr. Ann?
Here, just to push a little more what was said. So from what you're talking, Gary, we really need to think as a real-time reciprocal interface between the empirical and the agent-based model. If we want to move to the precision approach within the type of domains that we are looking at, it says that we need to follow the path you are describing, if I understood correctly.
It depends on the time scales of the disease process that you're dealing with. So sepsis, which actually happens very, very quickly, the answer is yes, you probably do need fairly rapid turnaround between the observable states, this projection, and the imputed actions. Again, things like cancer probably don't need to do it that frequently, right? Because biology is slower, chemotherapy agents take a longer period of time. Right, be operating on weeks scales or month scales with respect to determining tumor host responsiveness to a particular chemotherapy regimen or whatever. Right, so I think that thinking of it as a control problem gives you some idea about what the kind of time scales you have with respect to being able to do whatever it is you need to be able to do.
But how do you bring together those various time scales that are all operating within the same systems that you are trying to model?
Yeah, I think at some level, there's a level of pragmatism that comes into what is actually feasible with respect to your intervention space. Like, for instance, with the deep reinforcement learning we did, which worked, those are very unrealistic action spaces, right? You could check the state of the system every 6 minutes and every particular cytokine nearly instantaneously, and that's not doable, right? The genetic algorithm was a little bit more plausible because it said, okay, we're going to drug every 12 hours. Right. The point of these particular experiments is to essentially identify the boundaries of investigation. Because if you say that in order to control the system, you have to be able to sample at this interval, and you have to be able to interval in the degree of multimodality. Any attempt to coarsen that temporal scale will result in ineffectual control. That tells you the people who are doing sensor design use this, right? It drives the development of additional technologies to meet criteria that the simulations predict, which again, in some ways, is how microprocessor engineering occurs, right? They determine what the requirements are for the thing to actually work, and they develop the technologies to be able to meet those particular criteria.
Yeah, and I have a comment just on that point. I mean, there's going to be a fundamental difference between, say, a systems biology model and a weather model versus human, like the type of models that we're talking about when we're talking about human behavior and population dynamics and that. The two higher systems are primarily, physics related, and a lot of the uncertainty comes in stochasticity, interactivity of the different physical properties of the system. And we have another axis here that we're talking about when we talk about behavioral aspects of population dynamics, in that the rules of those systems are both highly stochastic and on top of that not well understood in terms of the underlying mechanisms that dictate overall exactly why humans do what they do. So the other axis that we have to sample, I would say, Lorette, too, is knowledge axis. So part of another sampling this is we're trying to take models that we're creating and incorporate new findings into that space in order to be able to update the understanding of the overall system, because what we do at this point, we do not have there's no way that we actually have a complete picture of human behavior. It's just, it doesn't exist in the scientific literature or at a mechanistic level. And so there's another axis we sample there, which is, people are gaining new insight, be true or untrue. I mean, there's always the fallibility of the insights, but we're seeking also to incorporate that knowledge into the model as we move forward. So there's another sampling there that's at a different time scale than the physical sampling of the system. It's the sampling of knowledge acquisition towards how the system works, if that makes sense.
Yeah, I mean, there are kinds of stochasticity that are intrinsic to these systems. There's aleatory stochasticity, which is intrinsic to the system, and Sean, with his quantum chemistry background, is familiar with that particular component, right? But there's going to be perpetual epistemic stochasticity, where it is impossible, at least at the scales that we're dealing with, to know exactly all the features of that system, all the interactions between components which you don't even know may exist, right? So I think building in terms that will account for that uncertainty, that again is matched to empirical data, is the best that you can actually do. The key is you have to incorporate particular uncertainty as stochastics is how you can come up with robust solutions. Because again, going to the idea of control, real world, you're going to have to deal with that uncertainty, right? In fact, controls can deal with those uncertainties. The control module that you're trying to engineer a system to deal with should be able to account for the fact that it has this aspect that is just uncertain. Therefore, so an example of that is the ability of the DRL and the GA to drive systems at the stochastic zone of the agent-based model. This goes to go back to the crisis of reproducibility, which is the translatability of outputs from fragile experimental systems, right? Any sort of control that is based on knowledge that is extracted from a very fragile, brittle system, it would be a brittle solution.
Yeah, they're positive that the modeling can help us understand the fragility of those results as well. As we're getting, I mean, this is a lot of that's a stupid thing. A big part of what we're working in the Hubble project is how do we have a lot of siloed efforts that are looking at sort of the same thing, why people do what they do, and how can we combine those. And so we should be cognizant of that. And I think there's a big role for agent-based modeling in that process in terms of synthesizing things and insight that one has from those studies into a place where one can combine them and understand how to interact and where, and just exactly what you're talking about, the reproducibility and understanding and design of new empirical studies beyond that to be gain more insight. So there's like a knowledge acquisition piece of this that's the axis that I was kind of referring to.
Yeah, I completely agree with you.
Okay, any other questions for Gary before we adjourn?
Very, very much, Gary. That was excellent and
My pleasure.
very useful.
And thanks for the conversation afterwards too.
Yeah, I really appreciate it, Gary. This was excellent. It was a really good inspiration for the type we're doing, and I hope that we can continue the conversation in the future, because it seems like there's a lot of work here, and it's really impressive the thought that you put into this that I think really brings these concepts home to people that have been in modeling for a long time. So really appreciate you taking the time to talk to us.
It's my pleasure, and thanks so much, and we'll be definitely talking more soon.
Well, thank you everybody for joining us, and we currently do not have another Bridge webinar scheduled, but we'll be sending out emails to let everybody know when the next and notifications when the next one is. And have a great day. Thanks.
All right. Thanks.